everyone, welcome to my YouTube channel. Today I'm gonna show you how to do EMG amplitude analysis. Especially I'm gonna talk about the mean, peak, and integral amplitude of EMG. So goal of this YouTube video is to make this plot. Like I said, mean amplitude of muscle activity in EMG and peak amplitude, and we're gonna find integral EMG amplitude, which is kind of same as impulse procedure. So we're gonna do pre-processing the data first, then find the muscle activation duration, then find the mean EMG in each muscle in each trial, and do the same thing but peak EMG. Then I'm gonna do integral EMG analysis. So I'm gonna show you how to do those all procedures by using MATLAB. So let's go to MATLAB. Okay, MATLAB. So I already did pre-processing the data and found the muscle on, off, as well as muscle activation duration. I mean, if you don't know how to do it, I made videos to show you how to do timing analysis of EMG and how to do pre-processing. So just go check my previous videos. So first thing I'm gonna do is make a plot to just look at overview of envelope I just created in my previous video, and I'm gonna plot it trial by trial. So let's run this. So this is first trial. As you see on the legend, blue means right bicep femoris, red is right semitendinosus, yellow and purple are just left side or left legs. So as you can see, it's kinda tough to interpret the data by just looking at this plot. And same for trial 2, trial 3, 4, and 5. And yes, this is just the overview. No comparison can be made, it's just the overview. So technically I said comparison, but without normalization you cannot really compare one muscle with another muscle or one trial with another trial. But this is just the overview, so I'm gonna show you another overview in muscle by muscle. So I'm gonna run this. So I showed you trial by trial, but this time this is muscle by muscle. So blue represents trial 1, red is trial 2, yellow is trial 3, purple is trial 4, and green is trial 5. So what you can see is amplitude is kind of same pattern. I would say just keep increasing. Amplitude is increasing as time goes by, and the duration is decreasing from trial 1 to trial 5. And this is only for right bicep femoris, and I think you can see kind of same, not same pattern, but like duration wise, the first trial is longest and the fifth trial is the shortest. Amplitude is a little bit different in this time. But for left bicep femoris, I think amplitude pattern is kind of same, maybe a little, I would say similar, not really the same, but duration wise it's same, I think across all muscles. So the first trial is the longest and the fifth trial is the shortest. And the right semitendinosus and the left semitendinosus have kind of same, it's tough to say, but first trial amplitude is a little bit lower than other trials in left and right semitendinosus, although I cannot find those patterns in left and right bicep femoris. But this is just overview, you cannot really compare. So let's go to deeper looking. So I'm gonna find the mean amplitude of envelope I just showed you. I'm just gonna use function called mean. I mean, all data is saved in a variable called move data, and you just extract the data because this is cell array. It's kind of tough to manage the data with the function, so like I just extract the data and save it into a variable called envelope, and just by using envelope I'm gonna find the mean one muscle by one muscle, and based on muscle on and off. I'm gonna just exclude everything not between muscle on and off. So I'm just gonna take only duration which muscles are active. Then I'm gonna show you the plot. So x-axis is trial and the y-axis is amplitude, and as you can see on the legend, blue is right bicep femoris, red is right semitendinosus, yellow and purple is just the left legs. Then I try to find a pattern across trials, but seems like trial 4 is the highest in amplitude across all trials. But other than that, it's kinda tough to see the pattern across trials. And again, I cannot compare one muscle with another muscle or one trial with another trial without doing normalization. But this is just the mean amplitude of muscle activity in EMG. Then what I'm gonna do next is find a peak. So I just change the function right here, max. Then I'm gonna show you the plot. Okay, so this time I cannot say trial 4 is the highest among all trials, but again it's kinda tough to see the pattern. But peak amplitude is used in normalization. So for example, you can take, let's say, peak value of right bicep femoris, and based on this value you find the normalized value on envelope data in right bicep femoris in other trials. So what you can do is take out all values of right bicep femoris like I did right here, create a variable called envelope and extract the data, and you just divide those values with peak value. You just pick one of them, this peak value, or this one, this one, this one, this one, and you just divide the values of right bicep femoris in other trials with peak value. I'm gonna show you how to do normalization in another video, but this is just amplitude analysis, so like I just show you how to plot the data mean wise, peak wise, and the last section is integral amplitude analysis. So I'm gonna define integral part. So integral is pretty much impulse, so amplitude times time, and I mean you can define the integral from muscle on to muscle off, but like I just wanted to know what's the integral value in 20% of muscle activation, 50%, and 80%. So how did I define 20%, 50%, 80%? So you just do multiplication, so duration I just found, or I just created in my previous video, times 0.2, which is 20%, and do the same thing for 50%, 80%. Then I'm gonna take all value 0.5 before the 20% of muscle activation and 0.5 second after the 20% of muscle activation. So in total 1 second of integral. I'm gonna show you the demo plot in a second, but pretty much same thing for the 50% and 80%. And I use a function called trap z. This function is really useful to calculate area under the curve. I'm gonna actually show you the plot, and you will see what I mean. So I'm gonna run this. Okay, so this is the envelope for right bicep femoris in trial 1, and this is the integral part or area under the curve part. So 20% is middle of this colored area, and I defined 0.5 second in the left and 0.5 second to the right. So in total it's 1 second because it's 2000 sample frequency, so 2000 data points represent 1 second. So 1000 plus 1000, 2000, so 1 second. And yes, so 20% of muscle activation. So this is muscle on and this is muscle off. So 20% is right here, and this is 50%, so middle of muscle activation, and this is 80%. It's kind of end of the muscle activation. Then I created the plot muscle by muscle. So I'm gonna run this. Okay, so this is right bicep femoris. So as you can see, the amplitude or impulse is increasing from 20% to 80% in right bicep femoris. I mean, yes, as you saw on the plot, like you could kind of guess, like amplitude is getting increasing from the beginning to the end, and you can observe same pattern in right semitendinosus and left bicep femoris and the left semitendinosus. Actually, left bicep femoris in trial 4 is a little bit different. Let's see, yes. So the 50% and 80% of muscle activation amplitude or like impulse are kind of same. It's kind of interesting. If you remember in the mean amplitude analysis, I think I have here. So trial 4 is a little bit kind of weird because its amplitude is all like highest in all muscles across all trials. So maybe he kind of changed the technique of Nordic hamstring curl in trial 4, but it's just a guess. And yeah, you see kind of abnormal pattern in trial 4 in left bicep femoris. I don't know, but it's just a guess. But it's kind of something interesting, and I try to find other patterns. For now, I don't know. I mean, I'm gonna put this code on my blog, so you can play around and maybe you can find something else. But as far as I can see, I just can say it's increasing. So pretty much that's it. Thank you for watching my video, and as I said, I'm gonna put this MATLAB code on my blog so you can download it and play around and find something interesting. So maybe next time I'm gonna do normalization or maybe frequency analysis. So see you next time.